Today is an important week for many reasons. One is that it's my favorite week of the semester. I love fermentation. I teach an entire class on it in the spring. One of the challenges is to squeeze all of that into 1 week. So that's reason number one. The second reason is that so far in this class, we've been here for a few weeks, we have more or less cooked with heat, right? And so often when we cook, when we think about what cooking is, and Dave Waits even showed a picture from the Oxford English Dictionary showing the definition of cooking. So often when we think about what cooking is, we think about it as if it's cooking with heat. You grill things, you boil things, you add it to the griddle, you make a stew, bake things and so on, right? And we've talked about this in the context of two equations, right? So the week before last, we talked about it in the context of this equation, which basically said that if you have a hunk of stuff of the mass m, and you have a constant for that mass, then if you add this much heat, you increase the temperature that much. That was the equation of that week. Then last week, we looked at, on a molecular scale, if you did the same thing to a molecule. And what we saw for that was that phase transitions happen right as a certain energy that a molecule has, which is determined by the temperature, is equal to the interaction energy between the molecules. Right, so just to bring you back into this room of science and cooking, this is what we've talked about so far. If you think about it in terms of what heat actually does to food, you can. I'm going to be drastic. You can think of it as the sort of philosophical opposite to what we're going to talk about this week, which is kind of amazing to think about. When we add heat to food, we essentially kind of kill things within it, right? So you know that if you pasteurize milk, you do it by adding heat, and that kind of kills the bad microbes, right? You know that if you heat an egg, you essentially kind of kill the chicken that was supposed to come from that egg, right? You know that when one of you stuck your hands in oil and water, two of the most common cooking materials, you very quickly took away your hand from the water one, and if you had just kept it in there, it wouldn't end well, right? Heat messes with life. That's my point. This week is the philosophical opposite in terms of this week, we want to keep the microbes that can cook for us. We want to keep them not just alive, but we want to keep them happy. We want to kind of create a cozy environment for them so that they can do the cooking for us. So previously in the semester, we've cooked with heat. This week, we're going to cook with life. How is that for philosophy? Okay, that's as best as I can do it. Okay, so what we're going to do is, today, Arielle is going to talk about flavor and fermentation. On Thursday, we're going to go into more detail and kind of pull it all apart. In lab this week, you're going to make sauerkraut. Some of you already have. There is a sort of intro sauerkraut demo here that will start to get you thinking about what happens when you make sauerkraut. And then, as usual, I just want to kind of give you 3 or 4 things to kind of organize in your mind what's going to happen up here. This is going to be a pretty sort of theoretical lecture. There's a lot of information in there. And on Thursday, we're going to pick some of it apart. But kind of some things to keep in mind. As always, see if there are sort of 3 facts that stand out to you that are like, whoa, I had no idea. That is so interesting. Make note of that, because that was your curiosity that sparked you. And you should follow your curiosity. So that's number 1. Keep an eye out for 3 fermented foods. Pay special attention to the demos. And then I've actually added a number 4 here. And we're going to talk more about this on Thursday and in lab. But think about why salt is so important in fermented foods. Okay. So with that, I'm going to introduce Dr. Arielle Johnson, You look startled. So Dr. Arielle, she's a flavor scientist. She actually has a PhD in flavor chemistry. So she's been thinking about flavor for a long time. She is also a gastronomy and innovation researcher. She consults for a lot of companies that sort of want to take flavor to the next level and doing it with fermentation. And she's also a co-founder of the Noma Fermentation Lab. Who's heard of Noma? Yeah, so Noma is a restaurant in Copenhagen. It was kind of for many years, and I think maybe still it's one of the, it's like in the top 2 or 3 of best restaurants in the world. What they're famous for all kinds of things. One of the things is for kind of bringing out, using local ingredients and kind of emphasizing Nordic cuisine. But one of the key ways in which they do that is by fermentation. So they have a long tradition of using fermentation as a way to increase flavor. They have, in many ways, kind of sort of shaped the discussion about how fermentation has developed over the last 10, 15 years. And Arielle was there as it all started. And we're going to hear some part of that story. I actually asked her to give you a little bit of a background of how she ended up where she is today. Because that's something we're thinking about, like what to do next, what's going to happen. So you'll get that. And with that, I'm super excited. I've been wanting to invite Arielle for years. So this is super exciting for me. Yeah. Okay, so let's welcome Arielle. One more time.
Hi. Hello. Oh, yeah, great. Hi. So I'm going to talk to you guys about 2 of my favorite things, which is fermentation and flavor, and using fermentation to create flavor. So hi, my name is Arielle. I have a PhD in flavor, and this is me at age 3. Playing with my food, and I kind of have not stopped since. I actually grew up in the area. I'm from. Oh, thank you so much. I grew up in Brookline, so it is nice to be back. Yeah. So how did I end up coming to stand here telling you guys about stuff? I did a bachelor's degree in chemistry at NYU, and at the time I started getting really excited about. Well, not just cooking, because I'd been excited about that for a while, but wow, there's a lot of stuff that science has to say about cooking and cuisine and gastronomy. So I did a minor in food studies, which is sort of more anthropological, and did an independent study kind of thesis project on food chemistry, since there isn't really a lot of food chemistry departments outside of universities that have food science. So I was really interested in cooking and researching cooking, and do I want to become an experimental chef, or do I want to become a scientist that works on food? The science thing won out. I guess thankfully for anyone who would have had to have me in their kitchen during service. But so I knew that I really wanted to do chemistry, and I really wanted to do cuisine and gastronomy and questions about food that weren't just food processing. So I reached out to a lot of different programs and was just open about that. And most food science programs at the time were we don't really do that. We kind of do more serious stuff with the food industry, processing and stuff like that. But UC Davis in California has both a really excellent wine department, so they have an entire department of viticulture and enology dedicated to understanding wine, I guess, from the vineyard through to the glass. And they have this, they wrote back and said we have a kind of grab bag graduate group called the Agricultural and Environmental Chemistry grad group. And, some people there do atmospheric chemistry, and some do water quality. But there's people that study, wine and flavor and stuff. So kind of if you can make it work, we're not going to stop you from doing that, which is sometimes the closest thing to encouragement that you're going to get for something that you're really excited about. So I kind of dove right into that. And they didn't stop me. It worked. I got my PhD. So so sometimes I get asked okay, so you do food, are you a chef? Or oh, you're a scientist, so are you at a university or something? I am kind of really neither of those things. I don't really like to do, or I'm just personally, I just figured out about myself that I'm interested in what I'm interested in, and I try to find ways to do all of that. But for the most part, that's a combination of doing, actual, rigorous publishable research, various things, fermentation, flavor perception, how food labs work, but also applying that knowledge and working collaboratively with people who are doing applied stuff with food, namely chefs, bartenders, that kind of stuff. So I've worked with a lot of restaurants on research and development and innovation, Noma, as we mentioned, some other ones, Momofuku, Minibar, Mena, unfortunately, just closed, but hopefully we'll do something else. But and another component of that for me is because since we know that cooking and science is a thing, but there's not there's this course and there's a few other resources for it, but there's not a huge amount of resources. So I work either writing articles about that or working with TV shows like Good Eats on the Food Network to bring a really rigorous understanding of flavor and chemistry into understanding cooking. So I guess you could say I'm just obsessed with all the things that science and cuisine have to say to each other in any direction. And my main medium and focus is flavor. This is some of the trouble that I've gotten up to with that. So the topic of the day, fermentation. And fermentation is a biochemical process. I believe you guys are making sauerkraut this week. That's your lab, so it's a very hands-on thing. The way I frame it for myself is collaborating with microbes to create flavor. So there's two parts of this. Obviously, there's the microbe part, but there's also the flavor part. And so I'm going to start with that because it's, to me, the most exciting and also kind of the least well-known part of the subject. So there we go. Actually, I just realized I could use this one. Can I? No. All right. What is flavor? So when somebody asks that, or I ask that of somebody, sometimes the definition that people give is flavor. If food has flavor, it tastes good. Either it tastes good or it tastes not good. So you don't really ask someone oh, how's the flavor of that dish? Well, you might, but most people don't. You say oh, does it taste good? And you can reply yes or no. There is a flavor industry, so flavor can be things like stuff that you add to Doritos to make it taste, highly palatable, as we would say. And then the kind of other picture that people tend to have of flavor is a very refined and articulate palate. So, can you walk up to a glass of wine and say something like that? And all of those are true, but none of them are actually entirely true. So I like to start from the basics. Flavor is a composite sense or a hypersense. It's not just one of our senses. It actually incorporates all of our senses, but mostly taste and smell. And it senses molecules. So these are some molecules that are part of flavor. Taste and smell are chemical senses. So something like vision helps you see physical objects that are roughly the same size as your environment. Hearing lets you get an idea of things that are further away from you because they send vibrations through the air that you then hear a sound. Taste and smell are for sensing incredibly tiny amounts of stuff that are totally invisible that have some kind of usefulness, whether it's to do with nutrition or not poisoning yourself or eating a balanced diet or knowing that this plant, oh, I smelled this plant before, it's tasty. Oh, there's not, and there's you can't see it, but you can sometimes sense down to 1 part per billion or below of molecules. So it's a very specialized and microscopic sense. So yeah, I think the 3 most useful things to know about flavor, especially for this, is that flavor is molecules. So taste and smell, chemical senses, you're always talking about molecules. If you're talking about flavor, flavor is taste and smell. Those both create perceptions from molecules. And lastly, that flavor can be created and transformed because it's molecules. If you create new molecules or transform existing molecules, you can literally create a flavor where there was none before, if you change them in the right way. And so, like, fermentation is a great example of this. Fermentation involves microbes, kind of going about their biochemical business and creating new molecules as a process, as part of that process. Those new molecules have flavors, so fermentation is really flavor creation on a molecular level. So besides just like being a cool thing, taste and smell being a cool thing, understanding them helps us understand what we're doing when we're doing fermentation and what we're tasting and smelling and where that comes from, and perhaps what we could do with that to take it in another direction. So let's start with taste. Taste is like the most obvious part of flavor. Usually if you are describing the flavor of something, you say that it tastes a certain way, like, oh, it tastes like raspberries, or it tastes like tea, or something like that. So taste is kind of the like foundation of flavor. There are 5 primary tastes that everyone agrees on. That's sweet, sour, umami, bitter, and salty. There are probably some other tastes, but it's a little up in the air exactly like what they are and how they work. So these are like the big 5 to keep in mind. We've evolved the ability to perceive these tastes mostly for reasons to do with like basic nutrition and safety. So we can taste, our perception of sweet is caused by sugars, which is a fundamental source of biochemical energy. Our sense of umami senses an amino acid called glutamate. Amino acids are the building blocks of proteins, so it's like a good sign that proteins are there, and we need proteins, because otherwise, we're made of proteins, otherwise we'll die. Salty senses sodium, which is an essential mineral. We use it as a neurotransmitter, and if we don't get enough of it, our brain stops working. Sour, there's actually like a few reasons we think that we can sense it. Sour senses acids, and acids could be the sign that there is like vitamin C in something. We need vitamin C as ascorbic acid. We need that to survive. It can also be a sign that there's some like good fruit, or actually that something is fermented. There's some thinking that humans have been fermenting stuff for so long that being able to taste the sour taste of fermentation is a sign that food that is not fresh has been rotted in a good way instead of in a bad way. And so the last one, bitter, that's kind of an outlier. We're basically attracted to sweet, sour, umami, and salty. Bitter, we have an aversion to. Bitter is for sensing things that could potentially poison us, so for toxic molecules. We taste with taste buds. That's on the left, that's a taste bud. Each taste cell has a receptor in it that can kind of grab on like a specialized shaped catcher's mitt to like one type of molecule, and then fire off a signal that gets carried to the brain. So you get a signal for umami, one for sweet, one for salty, etc. And there's basically one receptor type, one like shape of this catcher's mitt for each taste, sweet, sour, salty, and umami. Bitter is kind of a specialized case just because there's like so many things that are bad for us that we need to make 20 different shapes of receptor to be able to like catch all of them. So we get these 5 signals and with specialized receptors. You may have seen on the left a like taste buds map that we can taste salty on the tip of our tongue and bitter on the back, and that's not really how it works. We have taste buds all over our tongue. Of all types, just sometimes areas of greater density on like the tip and the sides, but anywhere on your tongue you're going to get all of those taste buds. So speaking of taste buds, we all make different amounts of them. We all like express different amounts of those proteins on our tongue. We have different amounts of taste buds, which leads to our first demo. You should have gotten a little strip of filter paper, and we're going to figure out how many taste buds we have on our tongues. If you could please lick it, and I apologize in advance to those of you who are going to find it incredibly bitter. It contains a harmless bitter molecule called phenylthiourea. This is like just informed consent. Okay, so can everyone who found it really bitter raise your hands? Okay, so like actually that's like about a third of you, and that actually tracks with the population. So if it's extremely bitter, you are what is called a super taster. You have more taste buds on average than everyone else. Is there anyone right now who's like, what are you talking about? This doesn't taste like anything. Can you raise your hands? All right. So you guys are on the like opposite end of the spectrum. You're what we call hypotasters or non-tasters, unfortunately. Oh, good. I'm glad. Awesome. I'm glad you could all learn something about yourselves today. Yeah, so if it tastes super bitter, you're what's called a super taster. You have more taste buds than everyone else. The remaining 75% of the population, and I'm sure you're feeling very smug about yourselves right now. If you can't tell, I am not a super taster. I am a non-taster. It's listed as a hypotaster here. And that just has to do with having fewer taste buds than average. But there are some benefits to being a non-taster. Since super tasters are so sensitive to bitterness, you often can't drink coffee or beer. Not that you should be drinking beer because you're possibly not of age. Anyway, bitter things are bad example. Bitter things, which can include coffee and tea and sometimes even cheeses, are often too intense for you. So often people who are picky eater or labeled as picky eaters into adulthood, you just have more taste buds than everyone else. If you're a non-taster though, or a hypotaster, since you have just generally less sensitivity than average, you can kind of throw a lot of stuff at your tongue and still have a good time. So you guys probably like lots of weird foods, or you're willing to try lots of weird foods and not find them gross. Again, spiciness also is probably slightly less painful to you. So that's good for if you're a non-taster. Fermentation's good for us because it can make lots of funky flavors, and we like things like that. Fermentation's also good news for super tasters because fermentation usually involves salt and sourness and often umami, and all of those things are good at masking and counteracting bitter. So regardless of where you fall on that curve, fermentation can be great for you. So second part of flavor is smell. Smell is for sensing volatile molecules, and in this diagram, the way we do that, volatile molecules are molecules that are basically light enough to become a gas. They either are a gas all the time or some of the time. If you can smell a flower from far away, its volatile molecules are drifting through the air and going to where you can sense them. So olfaction, smell. You take in air full of volatile molecules through your nostrils. There is a bit of tissue at the top of your nasal cavity called the olfactory epithelium that is full of smell receptors, which, like taste receptors, have grabby little specially shaped hands to grab onto molecules. Those send signals to your olfactory bulb, which is part of your brain, and then that kind of passes it to the rest of the brain. So, it's very, very basic physiology of smell. Smell is awesome. I'm very biased. I'm a flavor chemist, but smell is really my wheelhouse. It's for one, it's responsible for everything in flavor beyond sweet, sour, salty, bitter, umami, and spicy. So if you think of fruity or roasted or smoky or funky or vegetal or the flavor of orange or coffee or cherry, all of those come from smell. So most of the color and detail in the world of food and flavor comes from smell. Again, smell comes from volatile molecules. And whereas for taste, we have these 5 main channels and usually 1 receptor per taste. We have 400 different types of receptors for smell, so we have a much more diverse set of those. And that actually does not limit us to 400 different smells. We actually combine signals from those receptors to make much more complex things. So there's probably 40 billion different potentially smellable molecules. This was some recent research where they did a bunch of chemistry calculations. And then because of our flexibility with creating stuff out of these signals, it's been estimated that there are 1 trillion different smells that we can smell. So that's that research. Smell is sensed basically through patterns. So taste gives you like a very, clear signal that like, oh yes, this is sweet, this is sour, there's a salty thing. With smell, we actually, we perceive smell kind of in a way neuroscientists call odor images is how we like store and perceive smell. So it's actually like kind of a bit more like vision than like taste in that way. And basically, we have 400 different types of receptors, and like each of those is willing to grab on to like a bunch of different molecules. And any molecule that you could smell is willing to like bind with several different types of receptors. So rather than generate one really clean signal, you get this very complex one that's stored on the olfactory bulb, like literally like a spatial image. So like each of those glomeruli, it's called, is attached to one type of receptor. And the smell of any molecule is stored not as like one singular signal, but like basically a map or a barcode, which makes it very difficult to study, but very fun and cool if you like weird stuff. So at this point, it's like, all right, yes, smell is cool, but like it's not always obvious how smell is a part of flavor, because we think about smell, we think about like, holding perhaps a plate of food in front of our face and smelling things that come off of it, but like that doesn't really play in when the food's like actually in your mouth, does it? Well, actually, it does, because we have two ways of smelling, and the exciting one is called retronasal olfaction, and we're going to demonstrate what that is with jelly beans. So if you guys could open your packets of jelly beans and pick one out, especially like anything that looks to be like a fruit flavored one would be especially useful for this. Oh, actually, can I have one too? And I'll do it with you. I like green one. Okay. Oh, great. Perfect. Thank you. Okay. So don't eat. Well, before. Okay. So take your jelly bean that you've picked out and hold your nose. And while you're holding your nose, put the jelly bean in your mouth and start chewing on it. And like, personally, I can taste like sweet and sour right now, and it's not very exciting. So before you swallow, let go of your nose. What does it taste like? Yeah. So I just asked, what does it taste like? Really, what does it smell like? So when you unpinch your nose, you actually allow... Oh, there we go. Flavor molecules that are in that jelly bean to float up the back of your throat into your nasal cavity. In other words, the back door, thus retronasal, backward smelling, and activate those smell receptors just as if you were sniffing something in front of your face. You might notice that, like, okay, when I unpinch my nose, sorry, you're like, yeah, I can get more flavor, but it feels like it's happening in my mouth. Your brain is tricking you. So, sorry. So, yeah, like huge amounts of flavor come from smell, but to like, we think to like, encourage, so that evolution encourages you not to overthink it. It kind of like takes that sensation and relocates it to appearing like it's coming from inside your mouth. So, yeah, your brain is playing tricks on you, but smell is this big part of flavor. So, flavor is molecules, like we said. We can taste sugars, acids, minerals, amino acids, and poisons, essentially. Smells, there's like a gigantic number of categories of things that we can smell, but some of them, like in nature, are like plant weapons, like the smell of garlic or horseradish. Actually, the smell of most herbs and spices is intended by those plants as a chemical weapon. We just enjoy it. Signals also. What we're most interested in is the microbial byproducts. So, like the funky smell of sauerkraut or the smell of vinegar are molecules that we can smell that are byproducts of fermentation. So, creating new molecules. So, we can smell the molecules that are already there, or we can have some fun and make them. So, making new molecules is really just about making or breaking bonds between atoms. And there's plenty of ways that we can do that. We can use chemistry or heat to make and break bonds. We can use enzymes. We can also use biology. And microorganisms or fermentation is a great example of that. And so when I say that, like, making or breaking bonds makes new molecules that have no new flavors, so this is the drawing. This drawing is of a protein. It's an unfolded protein, so usually proteins are folded up into a little ball. But in this case, a protein is just a long chain of amino acids. And that protein, most proteins, don't have any flavor. But that doesn't mean that their components can never have any flavor. That, like, oh, when you have a protein, no flavor ever. Because if you cut the bonds between those amino acids, you generate free amino acids. And one of them in particular, glutamic acid or glutamate, is our taste molecule for umami. So most proteins are like between 5 and 10% glutamate. So essentially, anytime you see a protein, you can think of it as a potential source for umami if you know how to transform it. So it doesn't currently have any flavor, but by changing it chemically, we can create flavor. So as I mentioned, heat is one way that we do this. On the left, we have like the process of burning wood. So like lignin, that big molecule, is what makes wood woody. And when you heat it up and burn it, you just basically knock some of those subunit shapes loose. And when they are detached from the larger molecule, they smell like cloves or vanilla or smoke itself. So when you smell smoke, you're smelling a only partially transformed like little bit of wood that's been knocked off. And on the right is the pathway for the Maillard reaction, which you may or may not have heard of. It is what makes things taste toasty or brown. So baked bread, roasted meat, roasted chicken. It's all like heat-driven reactions between things that aren't very tasty, and then heat makes molecules that do have a flavor. But biology, fermentation, that's what we're talking about, focusing on today. These are just a small survey of some of the molecules that are not found in ingredients until you ferment them. So fermentation will create things like acetic acid, which smells vinegary. It can also create lots of like fruity flavors. Ethyl decanoate here, that's an ester. Lactic fermentation can create acetone and diacetyl, which smell like creamy and buttery. So actually most butter until like the 20th century, the milk was actually fermented with lactic bacteria before it was churned. And so the flavor of butter is actually like the flavor that the bacteria bring to butter. So it's not actually like from the cow, it's from this microbial process. So this is actually the same molecule that they use for like movie butter popcorn, just sort of faked. And then flavors like blue cheese, hexanoic acid smells more like cheddar, the flavor of honey, some of these aldehydes. Basically, with fermentation, you're creating chemical complexity, which creates flavor complexity. So let's figure out how we do that. So when we talk about fermentation, there's kind of like different ways that you can frame it, or like I like to think about most ideas as different ways that you can frame it and what you can do with that. So from a kind of like culinary and like historical context, you can define fermentation as preserving ingredients by deliberately letting bacteria and fungi grow in them, because that's what you do. If you're being like a strict biochemist, you might define fermentation as the anaerobic process of converting carbon to energy performed by, among other organisms, bacteria and fungi. This sort of strict definition leaves out some microorganisms that, like, in practice we call fermentation. So it's useful to know the technical definition and also how that differs from the practical definition. And then there's the way that I like to think about it by bringing both together, which is transforming ingredients with microbes, including creating new flavors. So fermentation probably started as a preservation technique. So if you just leave like raw vegetables to sit for 2 months, probably can't eat them afterwards. But if you salt and ferment them correctly, you have a food that can last you through the winter or beyond past the harvest season. So it's a way of like extending shelf life and seasonality and preserving stuff. But of course, a fringe benefit is that it is delicious. Sauerkraut, I mean, I like plain cabbage. Cabbage is good, but like sauerkraut is more delicious than cabbage. And so if you have a like only raw or only like untransformed foods diet, fermentation adds a bunch of like flavor and excitement that you like really wouldn't get otherwise. So the main microbes that cuisines use for fermentation, we've got like bacteria and fungi. So we've got a little like map. Bacteria up here, fungi down there. Fungi can be molds, like koji, filamentous fungi, or yeast, single-celled fungi. And bacteria, lactic acid bacteria is the biggest one. That is what will be making your sauerkraut when you make sauerkraut. There's also acetic bacteria, which we use for vinegar. And besides categorizing as bacteria and fungi, some of these things need oxygen to do their stuff. So acetic bacteria, filamentous fungi. And on the other side, we have things that don't need or don't like oxygen. We call them anaerobic. Besides just kind of being like a cool fact to know, knowing like what each type of microbe that you might want to use like needs in its environment helps you kind of like coax them along to stuff. Fermentation like can seem kind of like, oh, it's a trendy thing or whatever, but I guarantee you, even if you're like not into it or you think you're not into it, there is something that you love that you can only have because of fermentation. So bread, beer, yogurt, even things like coffee and chocolate have to go through a fermentation process before you roast them. If you try to roast coffee or coffee beans or chocolate, cacao nibs, without fermenting them first, they're just like, is not the stuff that's needed for heat to create flavor. So you have to put it through a fermentation process so that the microbes can do a transformation and then create things that then, when you heat them up, create the flavors of coffee and chocolate. So fermentation is really like absolutely key for so many flavors in cuisine. And, again, like just using really, there's many species in each of these categories, but it's like not that many categories of things, and we can create tons and tons of stuff from them. People have found ways to like adapt them to their local ingredients all around the world. Balsamic vinegar they make in Modena, Italy. That's a grape-based vinegar. They also make vinegar in Japan. This is from a vinegar brewery I went to in Kyoto. In that case, it's made out of rice. So, essentially, like anywhere that you have alcohol, which is most places, you can make a vinegar out of that. These microbes are like pretty widespread, almost universal, or global, but by no means like uniform. They're very adaptable to like local circumstances. And one fermentation I'm like particularly excited about has been called the wonderful world of the grain molds. In other words, things like koji, nuruk, meiju, molds and other fungi that do fun stuff to grains and legumes. The iterations that you see are things like miso or soy sauce or doenjang, but they all start with the same or similar molds and other filamentous fungi. So just because like we're using something like Aspergillus oryzae or like Rhizopus oligosporus, I'm just name dropping. Sorry, it's not always going to look the same. Depending on like what you want to use it for. Like this koji, this is mold grown on a soybean, and that's going to become like one particular type of miso. This koji is going to become sake, which is going to become rice vinegar. And then these are both Japanese. And then in Korea, meiju is the well, this is called meiju, and you get it by boiling soybeans, making a block, and then letting it sort of naturally inoculate with these fungi and some bacteria. And so there's like this place that I visited, they have just like a whole room to hang meiju and let it get ready to make doenjang, which is like a soy paste, and ganjang, which is soy sauce. And but if you want to use that mold for something else, for example, like rice wine, you will grow it on like rice or other grains instead and make nuruk. So just a few microorganisms can make like lots of different shapes and flavors of things. Yeah, so this is like two versions of one of those. We have miso, which started with those green little soybeans, and then the meiju on top went to this doenjang. So both soy pastes, but different vibes, which is fun. Okay, so I've made a lot of miso. It's fun. It's delicious. It has lots of umami flavors, and a lot of what I do is like, especially with fermentation, is like looking at traditional processes, trying to figure out like what the underlying biochemistry is, and then like from that thinking about how I can use that. So like I always try to start with how things are like traditionally and culinarily made, because they're usually more delicious that way. So something like miso, you'll start with like rice and a mold called Aspergillus oryzae. You'll make something called koji out of that, which is full of delicious enzymes. Then you'll add cooked soybeans and salt and let it sit for a long time. And so you'll go from salty boiled soybeans and sweet rice to wonderfully complex rice miso. So, thinking about it on like just a culinary sense, like maybe that's like, it's enough to just know that happens. I'm pathologically curious, so I'm interested in science of fermentation. So most fermentations involve salt. That miso involves salt. The sauerkraut you guys will be making involves salt. Oops. And it's kind of like a known thing, like, oh yeah, salt is a preservative. Does anyone know why salt is a preservative? There are no wrong answers, but just curious if you've gone over that chemically before. No? Oh, yeah, in the back?
Because it like breaks down cell walls
Yes. So it doesn't actually break. Very good answer. It doesn't actually break down the membranes, but yes, it's. No, it's okay. Yeah. that was great. But yes, so salt does something or drives something called osmosis, which basically nature hates an imbalance. So if you, for example, have a bunch of we can do the demo cam. Okay, if you have a bunch of shredded cabbage. Like this, cabbage plant cells have water in them, and by slicing it up, we've like exposed some of those cells, and so they're like a little bit wet on the surfaces. If we add salt to that, this is about 7% salt, I think. Okay, and mix it together. Like nothing happens right away, and this is like. Eventually this will ferment. We have just salty cabbage. But this is obviously not sauerkraut yet, and there's many steps that it takes until we get there. One of those, if we're like thinking about it as cuisine, is the creation of brine. Oh, there we go. Okay, so this is this same cabbage that has been salted probably about 4 or 5 hours ago, and we're already seeing changes. We're not really seeing microbial changes now, but we're seeing physical changes, and one of those is that it's giving up this brine. I can maybe you can squeeze it out. That would be cool. Okay, yeah. There's so much brine, so much juice from inside the cabbage that we can actually squeeze it out. Okay, a little bit of cabbage in there too. So what we have done is use the salt to do osmosis. The salt outside the cabbage leaf, or by putting salt outside the cabbage leaf, you're essentially creating an imbalance where there's more salt outside the cabbage leaf than inside the cabbage leaf. And to correct that imbalance, because nature hates an imbalance, water, which is a bit easier to move than salt, will kind of like rush out of those cells, collapse down the cabbage leaf until it's sort of floppy, and then create a brine by like dissolving the water that's around that. So there is a chemical component like to what is happening with sauerkraut. Sauerkraut would be like not really the same thing without like floppy texture and brine, but the exact same thing happens to microbial cells as happens to cabbage cells. So when we, much like this piece of cabbage, if you put salt outside of a bacteria, the water inside of it will rush out and the bacterial cell will kind of collapse and die. And fortunately for us, there are some species of bacteria that have developed the ability to counteract this. They can actually like tolerate some levels of salt. And those are, among other things, lactic acid bacteria. So by adding salt to this, we have not only like created an environment that's hostile to most spoilage microbes, including like nasty molds and not so good bacteria, but created kind of like a filter so that only lactic acid bacteria, which we do like, can grow and thrive in there. So like the salt has this functional role of creating a brine, and we need the brine for the right texture. And for the microbes to live in, but also like the salt itself is a way of selecting what microbes are going to grow there without necessarily having to like buy a packet of lactic acid bacteria and add it to it. There's lactic acid bacteria all over your hands, probably all over every surface, definitely all over the cabbage. And just by adding salt, a very traditional thing is just, we just add salt and that preserves it. Well, by adding salt, we're actually making a quite sophisticated selection of what microbes will actually grow. I think of this as a cozy environment, as Pia mentioned. It's really a collaboration with microbes. So, this will become sauerkraut, if we kind of just, leave it alone. So, we don't have to brute force anything. We just have to, make an environment that's, a nice spot for microbes to live in, and, they'll do their thing. So we give them a cozy environment and a source of food. They give us natural preservatives and tasty waste products. And those preservatives, the tasty stuff, there's nothing that we have to do to make them make flavors. The flavors are molecules that are just byproducts of the microbial metabolism. So metabolism is just the sum total of all the breaking down and building up chemical processes that a living thing does as it goes along. And so with this example, this is a hypha. It's the little filamentous end of a mold, like that koji. And the way that molds get food out of their environment is they produce enzymes on the inside of their cells and then sort of just push them out into, for example, a piece of cooked rice that they're growing in. And those enzymes will that's part of their metabolism. They will break down the stuff that's around them. And in some cases, that's proteins. So some of those enzymes are called proteases. We have this nice formal way of naming enzymes where we just add ase to the end of whatever it breaks down. Proteases break down proteins and makes free amino acids that the mold can then use to make its own proteins. And then in the case of molds that grow on rice, they're surrounded by starch molecules, and a starch molecule is just a very long chain of individual sugars. It can be long and linear, it can be kind of bushy, but it's just glucose, just a big molecule for storing sugar. And partially so that things like microorganisms can't get at that sugar. But if they have amylase, which is derived from the Latin name amylum for starch, they can just kind of snip between the bonds on that chain and make, in this case, that's maltose. They can create glucose. Create sugars. And the mold itself can use those sugars to ferment stuff. But they're also sweet. So microbial metabolism is literally the same thing as microbial flavor creation. So it's just the microbes chill out and do their thing, and we get tasty stuff out of it. So to go back to some of those molecules, some of these things are the direct product of a process like that. For example, Actually, I don't think any of those are. Sugar and amino acids are good ones. But some of these, like, besides just like creating food and then eating the food, there's all kinds of like other things that go on, like recycling the fatty acids that go into cell membranes and stuff like that. And so like all of those various many processes will just like create byproducts. So maybe you're like sawing off some fatty acids from a like fat molecule. Well, like some of them are going to have flavors, and maybe you are kind of trying to store them for later because organic acids are kind of like toxic slightly. You might just like stick an alcohol on them and create an ester. So an ester is just an organic acid plus an alcohol, and then you've created like a fruity flavor just by the accident of what it's usually going about doing. And so that's all like fun and heady and feels very like mystical and nice. But I'm really interested in like having theory but applying it to practice. So like experimenting with fermentation. And I'm going to try to blast through these so we still have time for questions. So we can go back to the idea of the miso making process. This is something that miso is a fermented soy paste, and the enzymes in the koji that you grow first act on the soybeans and create free amino acids and fermentable stuff and lots of complexity. So this is something that, again, like I've worked with a bunch of restaurants that are interested in fermentation. Miso has been like a big one, and besides just like making miso the way it's already been made, you can kind of like abstract this a little bit. So like, oh, okay, well, like, I know I'm adding soybeans, and, like, what does that do? Well, some carbohydrates, but also, like, mostly proteins. And, so I have this, like, rice koji, and why do we need to use that? Well, the protease and amylase enzymes and also the starch and sugar that it makes. We need a little, like, liquid to keep everything, from becoming too dry. And we also need salt, and that's going to inhibit molds and spoilage microbes, make it taste salty, but also select for some salt-tolerant yeast and lactic acid bacteria to like carry on some of the interesting fermentations that are going to keep happening. So if you kind of think about that abstractly as like, okay, well, I need like the right mold and salt and a starch source and a protein source. If you know what things have starch and protein in them, you can kind of apply it to anything. So at Noma, we made a product called Piso, which used Danish yellow peas instead of soybeans and barley for koji instead of rice. This is like a Danish variety of barley. You can actually, there are some barley misos from Japan, but this is like Danish barley. So this is like a product that is like miso. It has umami flavors, it has salts, it has some of these fermented flavors, but kind of like we talked about towards the beginning. It's a process that is like applicable to like the correct molecules if you have them in your environment, and will create something that tastes different but kind of in the same genre. So like if you like treat this as sort of a flexible process, you can kind of like if the ingredients have the right molecules in them, throw anything at it. So like protein-rich buckwheat and some charred onion for extra carbs, or okra seeds and sorghum, cowpeas and millet, pumpkin seeds and rice koji. Like each of these creates something that's like miso, but like not exactly like miso, and kind of expresses, like through the sort of like interaction of the microbes that are growing and their environment. For example, like pumpkin seeds would be their environment. Creates like different metabolic byproducts that create different flavors, and so you have like a product that like fulfills the requirement of tasting umami, but also tastes kind of like nothing else that you've tasted, which is fun for a chef. We've also like used this to kind of like address the food waste problem, which obviously is a huge problem. Fermentation is not going to fix it, or all of it. But if you think about food waste as like, when you say the word waste, it's like, oh, okay, so it's like garbage and not good stuff. But you can rethink food waste as long as you haven't like let it sit for too long as like, oh, well, it's just like the part of the food that I kind of like cut off and I'm not using. So like maybe it actually just, has much of the same properties as the stuff that I want to work with. And if I think of like its wasteness as an opportunity instead of as a problem, then you can actually like create something new out of it rather than just kind of having to deal with it. So like spent beer malt has some like residual starch in it, so that can go into like a malt soy sauce. I mean, leftover egg yolks, like not every home cook has leftover egg yolks, but if you're making a lot of like meringue or something, you have all these egg yolks. We made this like egg yolk miso out of it because they're very protein rich. If you are serving a lot of squid, you might have some squid scraps that are not nice to eat on their own, but like it's a great squid that you sourced because you're a good restaurant. Well, you can make squid garum. Garum is basically fish sauce. It's actually the Roman version of fish sauce. But yeah, protein to umami. And then, besides like umami-rich pastes, you can also look at different sort of juicy vegetables as the source of delicious-tasting fluids that you can then, if you do the right tricks, turn into vinegar. And so, like, if you're willing to kind of like be a little flexible and rethink how you look at things through like a molecular lens, you can actually like create opportunities where there were once problems. And yes, question. Oh, like in Italy? Not as garum. There's a product called colatura that you can find. I'm not actually sure where in Italy it's from, but it is essentially like anchovy garum. So the thing called garum has not survived, as far as I know, into like modern Italian culture as garum, but there are some places where they do like heavily salted anchovy products, so and call it colatura, and it is like quite similar to like anchovy fish sauces from Southeast Asia, because it's like basically the same process. So we kind of got really obsessed with that, so we built a fermentation lab. But the whole reason to do that, besides like, oh, it's good to have a place to store everything, is that like, if you're setting up a, if you're like thinking like this and being like, oh, I can put like different ingredients into this to get a flavor, you can also kind of like, the environment is often just as important as the starting products for getting the flavors that you want. So, in the pink here, we have things that microbes need to eat, and then in the blue, aerobic, anaerobic, hate salt, tolerate salt. These are all things that you can do, manipulating the environment to get like a more precise result. So, besides just like looking at a product and being like, oh, I can substitute stuff into that product. If you want to get certain flavors, you can like think about reverse engineering where they come from so that you can like set up the environment to be the correct one too. So like thinking, both forwards and backwards about these things. So, I mean, that's basically all that this was. It was like a bunch of incubators that we made out of shipping containers. And we had to like literally like design the like heating and wiring system ourselves. Those are like little heat controllers. And with that, like, basically made out of, the controllers were probably the most expensive thing in there. We had like all these customizable environments to like get the flavor, that result that we wanted in a more like precise way. So, you kind of like stick to what has always been done, or take matters into your own hands, I guess, is the takeaway from that. And just like very quick, not plug of my book, but if you're interested in any of this flavor stuff, I got to the point where I was explaining stuff like this over and over again to people and being like, wow, I wish there was a book I could give you that explained this. So I'm writing that right now. It'll be out next year. And if being able to think about fermentation this way is interesting to you, there's lots of other ways that you can kind of look at the science of cooking as a like tool to apply, not just to understand. So thank you. Thanks for being a great audience and listening, and I'm happy to take any questions or.